Hello, my name is Hannah Flaherty, and I'm a junior studying medical microbiology with a minor in public health. I've been researching independently under the supervision of Professors John Bucci and Semra Aichor, looking at sediment samples from the Stellwagen Bank National Marine Sanctuary for biosynthetic gene clusters encoding for chemotherapeutic compounds. There was a great need to understand BGCs encoding for secondary metabolites from marine-derived species, as these organisms represent a biodiverse source of natural products. There are over 34,000 natural products discovered by researchers thus far that come from marine organisms, and the Streptomyces genus of bacteria alone accounts for over a third of all metabolites, two examples being actinomycin D and daunorubicin. However, in order to continue the discovery, maintaining health of ocean ecosystems is critical, as these ecosystems provide more than half of the oxygen on our planet, more than 17% of animal protein consumed, and are partly responsible for regulating climate patterns. Protection of these ecosystems include relieving pressures facing marine biodiversity, including habitat loss, overfishing, and extractive activities such as mining, and the spread of invasive species. So for this project, our objectives were to quantify our sequenced hits that align with Streptomyces hygroscopicus strains, and then categorize our BGCs by metabolite class and evaluate for functional characteristics of these anti-tumor compounds. We did this by taxonomically evaluating our data that was collected during the summer of 2018. We then used the program JUMP to look at the relative abundance of Streptomyces within our sediment samples, and the bioinformatics program anti-SMASH to align our sequenced hits with known metabolites within the NCBI library. And we finally used the MI-Big Source library to classify our known clusters with chemotherapeutic properties such as cytotoxicity. From our results shown to the right is an example of an anti-SMASH result, and we were able to use these to yield similar biosynthetic gene clusters, and we categorized those by chemical class such as siderophore and polyketide synthases. We showed those by the pie tables to the left to show which biosynthetic gene clusters was represented in our data the most. And we compiled all of our data up into the data sheet at the bottom with various categories such as subspecies and species with similar biosynthetic gene clusters. From our results, we were able to determine that compounds such as desferoxamine, which is an example of a siderophore, have potential for chemotherapeutic drug discovery, and these compounds show similarity to other compound classes such as polyketide synthases that have acute toxicity. This project offers insights to the importance of categorizing and protecting the biodiversity of sediment microbial communities while promoting sustainability for the medical field and discovering new medical drugs. Thank you so much, and thank you for listening.